Enzymes are protein catalysts that help biochemical reactions occur in our cells. Because of the unique shape of each enzyme, they have the capabilities to form and break chemical bonds by reacting to specific substrates. Substrates are any molecule that an enzyme interacts with. They bind to an enzyme's active site, which is specific to the shape of a substrate, and specifically the cofactor within the active site either forces a chemical bond or breaks a chemical bond. The substrate and enzyme connection happens by an induced fit. This means that the enzyme can slightly change its shape to fit a specific substrate. Once connected, this is called the enzyme-substrate complex, which is very similar to how a memory foam mattress shapes itself around you. Enzymes don't change the level of free energy available in an interaction, but it lowers the activation energy, helping to reduce the energy needed to start a chemical reaction. Organisms are so dependent on enzymes that without them, biological reactions, which are energetically expensive to start, would just die. In other words, enzymes increase the efficiency of biochemical reactions within cells. Degradation is an enzymatic reaction where a substrate binds to the enzyme and two or more products are made from the reaction. The result is usually energy being released. For example, a digestive enzyme lactase helps to break down lactose, a disaccharide found in dairy products. Please keep in mind that enzymes and substrates don't have these geometric shapes. These are there to help you understand enzyme activity. Lactose sugar binds with lactase at its active site. This causes a change in the shape to the lactase enzyme. Because of this change in shape, it literally breaks the glycosidic link that holds the lactose together. What it does is it forces the electrons to be so far from each other in a pair that they just break off, and the bond is gone. This causes lactose to be degraded into glucose and galactose, which are able to be used by cells. Notice the enzyme also goes back to its original shape, so it can be used again to break down more lactose. Interesting note, if you are lactose intolerant, it is because you don't produce enough lactase enzymes to digest lactose sugars. In synthesis, an enzyme forces a chemical bond between two molecules by bringing unpaired electrons closer to each other to help make a covalent bond. In the case of ATP synthase, it takes the molecules ADP and an inorganic phosphate and forces a phosphoanhydride bond, making ATP as a result. This happens with an insertion of food energy, causing the enzyme to change shape to make ATP. In reality, enzymes do not work alone, but in metabolic pathways. Each enzyme takes a substrate from a prior reaction and makes it into different products. The final product is what the cell needs. These pathways rely on enzymes functioning properly. Some medical issues stem from faulty enzymes not working in a pathway, leading to more or less product made than normal. And this is also how some pharmaceuticals work, either working for or against certain physiological processes either within you or within a pathogen causing disease. Enzyme A performs a synthesis reaction which binds to two substrates into a larger product. These products become the substrate for enzyme B, which changes the shape of the substrate. Finally, these products are degraded into two final products. There are many factors that affect enzyme activity. First, enzymes that function in catabolic reactions to degrade substances. The larger the substrate, the longer it will take to catabolize. Enzymes are proteins, and like all proteins, they can become denatured or lose their original shape if placed in the wrong environmental conditions. Colder temperatures slow enzyme activity, therefore slow down metabolism, which is why we refrigerate our foods. Bacteria rely on enzyme metabolic pathways to carry out our life functions. When exposed to colder temperatures, it takes much longer to grow and spoil food. Warmer temperatures, oh thank goodness. Increase enzyme activity. And one reason your body temperature increases when sick is to increase the immune activity so that you can fight off disease. And this activity requires increased enzymes activity to make products needed to fight infection. If temperatures are too hot, though, it can denature enzymes. Pathogens exposed to fever for too long can be killed since the enzyme activity is outside of normal temperature ranges, causing enzymes to denature. However, if the fever gets too hot or too high and is not controlled, it can do the same to our own metabolic processes in our cells. So you see, a fever is like having guests over. It's great, but only for a little while. pH can also affect enzymes and denature them if exposed to a pH outside of normal ranges. In this chart, two enzymes, pepsin and trypsin, both target and break down peptide bonds in the proteins found in our foods. Notice that pepsin functions best in low pH. That's because it is found in the stomach, where it is exposed to hydrochloric acid. Hydrochloric acid helps to destroy pathogens in our food. Once food enters the small intestine, however, the pH is neutralized by bicarbonate buffers found there. Pepsin can no longer function at higher pHs, but trypsin, which also breaks down peptide bonds and proteins, can. Enzymes must be regulated based on the needs of the cells they operate in. Otherwise, too much or too little products can be made for a given time frame. One form of enzyme regulation is called competitive inhibition. This occurs when a similarly shaped molecule fits into the active site of an enzyme. In this case, the inhibitor and substrate needed to carry out an enzyme reaction are similar in structure, but the inhibitor reaches the active site first, and this typically shuts down enzyme activity. This is not necessarily bad. Perhaps the cell no longer needs more of the product and the enzyme needs to be turned off. However, we can use this competitive inhibition to our advantage. In bacteria, they create folic acid using metabolic pathways. Sulfa drugs are an antibiotic that mimics one of the substrates needed for an enzyme. When it works, the pathway shuts down. Bacteria don't get folic acid that they need, and the bacterium dies. Allosteric regulation is a reversible reaction that changes the shape of the active site, either activating or inhibiting enzyme activity. For example, let's say the enzyme displayed is part of the metabolic pathway used to make insulin. This protein gathers excess glucose in your bloodstream and stores it in the liver and muscles. Insulin production must stop after glucose levels are normal. So a regulatory molecule will bind to an allosteric site, which changes the shape of the active site, preventing the substrate needed in the production of insulin from working, thereby stopping insulin production. However, let's just say you eat a meal and your pancreatic cells pick up excess glucose in the bloodstream. The enzymes involved in the cell to help make insulin must now remove the regulatory molecule in order to activate the enzyme by giving it its original shape so the substrate can bind to it. Enzymes help organisms overcome the cost associated with being alive. But no matter what, energy is necessary for living things. But this energy needs to be made in the form of ATP, or adenosine triphosphate. This small molecule supplies cells with lots of energy to carry out life processes. The high amounts of energy lie in the chemical bonds between the second and third phosphates. ATP is made of a nucleic acid base adenine and a ribose sugar, as well as 3 phosphates. The bonds holding the phosphates together are called phosphoanhydride bonds. And when these bonds are broken, lots of free energy is available. Since ATP to ADP hydrolysis is reversible, organisms require chemical energy found in carbohydrates, lipids, and proteins to repair the bond between ADP and phosphate. In other words, food. And since both catabolic and anabolic reactions already use energy to perform due to activation energy to start chemical reactions, catalysts are needed. Organisms use enzymes to help lower this activation energy to help save energy by carrying out necessary life functions. And so now that we know how organisms are powered to do this important processes of continuing life, how is ATP made from photosynthesis and cellular respiration? In the next video, we will look, and I promise, is quite briefly, at this important process.